it's the kind of thing that we face a lot, actually, when we're trying to develop new crops. Okay, so say we want to come up with a disease-resistant plant, but we can't find a plant in the population or in any population that's resistant to that disease. So we have no genetic variation to draw. But we still want to go ahead and solve that problem. And so there are a number of ways that we've developed in the last century to kind of create or bring in new variation. So the first of those methods is breeding by mutagenesis. So does anyone want to take a guess at what mutagenesis is? So it is basically generating mutations in the genome. So this is a process that happens naturally. So this is where all of the natural variation that we've been exploring and talking about this semester, this is where it comes from. So it's from mutations that are induced randomly by factors in the environment. So things like UV light or chemicals come in and make mutations in genes which lead to genetic variation. But that's a pretty slow process. We don't have a million years to sit around and wait for it to happen. And so we actually have ways of inducing lots of mutations. And so the first one that was developed was actually using radiation to generate mutations. And so this was developed in the 1950s. And so this is kind of cool, actually. I wish they still sold these. So these are poppy seeds, which they blasted with gamma radiation to randomly induce mutations. And so you can buy these and then plant them out in your garden and see what kind of plants develop. So they're all going to be poppies still, but some of them might have mutations that make them taller, maybe change the flower color. Any number of things. So this is actually an approach that's not just used for sort of at-home science experiments, but it's used really widely for plant breeding programs. So it still is to some extent. And so this is the Institute of Radiation Breeding, which is in Japan. And so this is actually it's pretty cool. And so this is a huge farm field, and it's surrounded by a big concrete berm. And then in the middle here, there's a pillar that has a radiation source. So it's cobalt-60. It's a radioisotope of cobalt, which is going to be emitting gamma radiation out into the crops that are in that field. So whenever they'll treat the plants that are growing there, and then the pillar lowers itself back down into a little bunker, and then the scientists can come in and collect the results. So this is a product that was produced at this radiation facility. And so on the right are the original flowers. The radiation caused a mutation resulting in pale blue flowers. Which is pretty cool. So you may have eaten, you're very likely to have eaten products of radiation breeding programs. So this one is the Rio Red grapefruit. So this was developed in the early 1980s in Texas. And so they had a bunch of buds of grapefruit plants in a field with a radiation source. And they isolated this variety, which has kind of a redder, more juicy flesh. So this is actually one of the most common grapefruit varieties that's grown in Texas. All right, so you don't have to use radiation. In fact, it's not that common anymore because it is kind of dangerous to work with, right? So you can actually use chemicals. So there's a pretty commonly used chemical called ethyl methane sulfonate, or EMS. You don't need to remember that. You can just remember that you can use chemicals or radiation to induce mutations. So this company, BASF, has been using this chemical to mutate different crop plants and look for plants that are to an herbicide that it sells. So if you have a plant that's resistant to an herbicide, that means you can go in and spray your whole field. Your crop won't die, but everything else will. And so that makes it a lot easier for the farmers. And so I'll mention this again on Monday when we talk about the debate around genetically modified organisms. And so in most countries, plants that are developed through mutagenesis, either radiation or chemicals, are not considered to be genetically modified organisms, which I'm seeing some skeptical looks, which is good. And we'll talk about whether or not that kind of classification makes sense on Monday. You have a question? Yeah, so the chemical and radiation ones, where you're doing random mutations, are not considered to be genetically modified. But the ones where we're inserting whole genes are considered to be GM. So we can talk about that a little later. All right, so showing you some products of mutation breeding. So let's think about how this might actually work. So let's say I've got my flower sitting in my radiation field. Where and what tissue in the plant is the mutation going to need to occur for it to be useful for me in developing new varieties? So if I have a mutation in a cell in the leaf here, will I be able to produce more plants in the cell?
In the seed, or
Yeah. Yeah, so it needs to be in a tissue where the genes are actually going to get passed along to the next generation, right? So if you think about it, only the sperm and egg, the cells that have gone through meiosis, are actually going to pass genetic information on to the next generation. And so we need to get a mutation either in a seed, and so seeds are what's most commonly treated with that chemical mutagen actually, or in pollen or eggs. All right, so thinking about this kind of random mutation approach, can anyone suggest what could be maybe a problem with this approach?
You mutate the wrong gene for whatever reason.
Yeah. So we have no control over what gets mutated here. This is totally random. So we're just bombing the genome with chemicals or with gamma radiation and looking for offspring that have things that interest us. So we might get a change in the gene that's beneficial that we could use, better colored fruits, disease resistance, whatever. But we could also be getting other mutations in the genome that we don't know about that could